In this video, I'm going to discuss the activation of specific effector T cells in response to a viral infection. We're going to go through this video firstly by talking about the different types of major histocompatibility complexes and how they affect the specific T cells being formed. Next, we're going to talk about T cell activation, and specifically cytotoxic T cells, helper 1 T cells, and follicular helper T cells. And then we're going to talk about how those effector T cells support other immune cells against viral infections specifically. So let's get into it. So beginning with the different types of MHC complexes, class 1 and class 2. Let's talk about class 1. So mainly class 1 happens with intracellular antigens. If we're using a virus, for example, the virus would have already infected the cell and would be inside. So what happens next is the virus is ingested by a proteasome, broken down into peptides, and then that's sent over to the endoplasmic reticulum. And in the endoplasmic reticulum, it winds up with an MHC class 1 protein or complex, and then that gets shipped up to the cell surface where it can be recognized by other immune cells. In contrast, we have the MHC class 2, and that happens with very specific types of cells. We can refer to them as professional antigen presenting cells, and these would be our B cells, our macrophages, and our dendritic cells. And we're going to talk about dendritic cells for the purpose of this discussion. So let's say that this is all a dendritic cell here, and what would happen is it would sort of sample the outside pathogens. So let's say this is a virus right here. It would sample this virus, and it would engulf it through various different mechanisms that we can discuss later. But it would engulf it, and then it would end up breaking down the peptides of this virus, and it would add this major histocompatibility class 2 complex to it, and that would eventually be put onto the cell surface so that other cells could recognize it. Now, there's a lot of different forms of being able to engulf a virus and present it on an MHC complex. These are some examples. The cell could be virally infected, and that would just pretty much, this would only be for MHC class 1 types of representation. And just to be clear here, MHC class 1 antigen complexes, those are recognized by CD8 T cells, which are cytotoxic. And then same goes here. This is just another example where a infected dendritic cell transfers its infection over to another dendritic cell, and then that gets represented onto the MHC class 1 complex, and that's another one. A cytotoxic T cell would recognize that. And here we have examples of MHC class 2, and this would be where macropinocytosis of viruses or even just receptor-mediated endocytosis occurs, and then those could be presented on MHC class 2s, and those would be seen by helper T cells. Helper T cells would recognize these ones. Okay, moving on. So once a dendritic cell has decided to represent a virus on its MHC complex, that could be 1 or 2, but once that happens, this dendritic cell is going to migrate. They're going to migrate towards the lymph node, because this dendritic cell wants to tell other T cells what's going on, that they're infected or that they found an infection, and they want other T cells to check it out. So if we have another image. But if you look closely here, you can see that all these little blue dots, these are T cells, and they're just kind of observing the dendritic cell and observing if they can make a connection with it and sense which type of peptide or virus or pathogen is on the MHC. So if we look closer here, the dendritic cell migrates to the paracortex, where the majority of, this is the paracortex right here, where the majority of T cells are. And as you can see, this is a better look. You can see the T cells are just hovering around looking at the antigens presented by the dendritic cell. And if the right connection happens, then T cells can start to become activated. So how do they become activated? Well, there's a few steps. There's 3 steps specifically. The first step is I'm getting ahead of myself. So first, I just wanted to say that these T cells here, they transiently connect to the dendritic cells. That's how they're getting so close. So this transient connection is represented right here and kind of what's happening here. The T cell will have an LFA1 molecule, and the LFA1 molecule specifically binds to the ICAM molecule in the dendritic cell. And you can see that this isn't the strongest bond. It's just presented schematically here, but it's not a very strong bond. So the T cells just sort of get close, bond transiently, and then move on. However, if the T cell receptor on the T cell is specific, look at that, specific for this epitope that's presented on the MHC class 2, then they can bind, as seen here. And if they bind and the co-receptor here, CD4, engages, then that triggers a conformational change of the LFA1 protein, as seen here. We can see that it's now in that square conformation, and that's a tighter bond. So what happens here is not only is this connection strong, the antigen-specific connection, but also we get a conformational change over here with LFA1, which makes it a very tight bond. And with this tight bond, it helps the T cell keep going with its activation process. So the next step here is we already have the antigen-specific connection that's happened here, and then it sends and then it sends a signal, and this is the antigen-specific signal. But also, another signal has to happen. As we can see here, the CD28 molecule binds to the B7 molecule on the dendritic cell. And when that binding occurs, that's called co-stimulation. It's a co-stimulatory signal. And this is the next necessary step. If this step didn't happen, co-stimulation, then activation wouldn't occur, and the T cell would become anergic and eventually die in a few days. So it wouldn't be a useful T cell for this immune response. So what happens after co-stimulation occurs? Well, then the T cell starts to express high affinity interleukin-2 receptors, and that's these guys right here. They're high affinity. You can see that they're just the right shape for this cytokine here. And what is this cytokine? It is interleukin-2. So interleukin-2 is secreted, and then it is received by the high affinity interleukin-2 receptor, just like this. And this is an autocrine phenomenon. So the T cell releases its own cytokine that is then received by its own receptor, and this is an autocrine reaction. But also this interleukin-2 that's being released, it can also have a paracrine reaction. And basically what happens is once the T cell recognizes the interleukin-2 that's released, then it will begin to proliferate and differentiate. And this can happen in an autocrine fashion or a paracrine fashion, as in that it would make other T cells differentiate and proliferate. Okay, so that makes sense. And this is just sort of touching on the energy, where if co-stimulation doesn't work, then we get an anergic reaction. And yeah, so we're just looking at that. So just to summarize, we have activation when we have the antigen-specific complex that happens. Next we have survival, which happens when the co-stimulatory signal happens. And then we have the cytokines that are released, and then that leads to our differentiation, which is very important. Now, the different cytokines are important too to consider. So this happens to every T cell, these three processes. Now, depending on the co-receptor right here, this one's CD4 for example, depending on the co-receptor, that's what determines whether we're going to get a helper T cell or a cytotoxic T cell. If we have CD4, we're going to get a helper T cell. If we have CD8, we're going to get a cytotoxic T cell. Now, there's many different types of helper T cells. There's, well, at least 5, probably more, much more than 5 helper T cells. For this discussion, we're just going to talk about the follicular helper T cells and the H1 helper T cells. And the way to stimulate those is you get the co-receptor CD4+, and then it depends on which cytokine is released. And the cytokines are based off the environment that the reaction is occurring in, the specific pathogen that's being presented, and other circumstances like that, just so that the proper T cells are being formed for the proper type of scenario. So I just would like to be specific here for which cytokines make which one. But we have interleukin 12 here and interferon gamma that produces the helper T1 cells. And then we have interleukin 6 that produces the follicular helper T cells. But that has to be in the context where we don't have, I'm just searching here. Yes. So for the follicular helper T cells, we have interleukin 6, also interleukin 12, or 21, my apologies, can do this. And that has to be in conjunction with an absence of TGF beta and an absence of the ICOS ligand. So that's important too, to make the follicular helper T cell. Now, what does the what does the follicular helper T cell? Oops. What does the follicular helper T cell do? What's its function? And where does this happen? Well, the follicular helper T cell pretty much stays, for the most part, in the secondary lymphoid tissue, such as the lymph nodes. And it does that so that it can help B cells. The whole point of the follicular helper T cell is to increase the antibody affinity of B cells, also to induce class switching. What does that mean? Well, of the antibodies. So as we can see in this image here, we have a follicular helper T cell, and by secreting different antibodies, it is able to stimulate the B cell to switch its antibodies to various ones. We can see we have a multitude of different types of antibodies here, and it all depends, again, on the specific cytokines that are being released. For example, if the T cell, the helper T cell releases an interferon, then IgG production will be stimulated. If interleukin 4 is secreted, then IgE will be stimulated. If interleukin 17 is secreted, then IgG as well, a different form of opsonizing. IgG will be formed. And then if we have a combination of interleukin 10 and TGF beta, then that will promote IgA. So if we didn't have these follicular helper T cells, then that means we pretty much wouldn't have the class switching ability of B cells, which pretty much means that these B cells would be highly ineffective. So the follicular helper T cells are extremely important in the adaptive immune response as a whole. Now, the other T cells that we're concerned about here today, the cytotoxic T cells and the other helper T cells, they migrate to sites of infection. So they are activated within the lymph node and other parts of the body, and then they migrate via cytokines and chemokines. They follow that trail to the source of inflammation and infection. And how do they do that? Well, they follow certain chemokines, but also they start to express different cell adhesion molecules on their cell surface. And these are specific to damaged endothelial cells and other properties too, but we're going to use damaged endothelial cells as an example. Damaged endothelial cells specifically will they will secrete, or they will express something called VCAM1. And what happens is these cell adhesion molecules on the T cells, they will bind here. Not too strong, but they'll bind, and this will slow them down. Once they get to an area of inflammation, like this place, they'll slow down, and then they'll be able to provide their effector functions in this area for a substantial amount of time because, well, they're hanging around for a bit. So overall, let's just discuss the final functions of the different T cells and how they would help fight a virus infection. So we're talking about the cytotoxic T cells here, and for the most part, what they do is they see MHC class 1 presenting molecules, and they see that they're virally infected or showing abnormal signs of pathophysiology, and then they pretty much signal for apoptosis. And apoptosis is important here. We want apoptosis and not necrosis, because apoptosis is a good thing. It's good because we keep the virus inside of the cell where it dies, so that a macrophage can come and eat it later. And also, we're not spilling the cellular debris all over the place like we would with necrosis. Instead, we're keeping it nice and compact, because with necrosis, we would have systemic inflammation that would happen afterwards as a result of the widespread tissue damage that happens during necrosis and inflammation that comes as a result. So that's the main function of cytotoxic T cells, is to just kill the virally infected cells. And then we have our helper T cells, the Th1 specifically. They produce a lot of cytokines, and all helper T cells, for the most part, produce cytokines that just overall improve and increase the immune response on a systemic and on local levels too. But that's the main function of these helper T cells, is they improve the immune response in a variety of ways. Helper T cells, the H1 variety specifically, they'll release cytokines to macrophages, and then the macrophages, this will trigger them to be more effective phagocytic killers, and also they'll release cytokines. They'll release more cytokines, which will trigger other eosinophils, neutrophils, mast cells, and just other innate immune cells to take part in the fun of destroying this virus. And yeah, I mean, that's pretty much it. The cytotoxic T cells and the helper T cells, the Th1 variety, they both contribute to killing viruses and to the overall immune response. TC by directly killing the viruses, and Th1 by recruiting different cells to fight the virus and clean up the debris that occurs afterwards. And that's how it's done.